on with the program and go to our next speaker, Cristal Hill, that joins us from beautiful Baton Rouge, Louisiana, Baton Rouge, Louisiana, at Pennington Biomedical Research Center. And she is going to tell us about FGF21.
Okay, is that?
Yep, you're good to go.
Okay. Good morning, everyone. First, I want to thank the organizers for extending the invitation for me to share some of the data that I've been working on for the last couple of years in Chris Morrison's lab at Pennington Biomedical Research Center. So I'm a senior postdoc, and my major focal point of my research projects has been around the metabolic effects of FGF21. And so today I'm going to share with you a story to hopefully convince you that the interaction of FGF21 and dietary protein restriction could be something that we could look at as therapeutic mechanisms for improving metabolic health as we age. So first, let me just start off with this figure. I think we all are very familiar with this figure, that as we increase in age, we also increase the risk for these metabolic or neurodegenerative diseases. But you can also take this in the context of obesity. So increasing in obesity also increases the risk for these same metabolic complications or diseases. And so there are diet therapeutics that have been established and that they actually defend against these age-related decline in metabolic or age-related illnesses. Most of we are very familiar with CR and fasting, but we really don't give much recognition to the reduction of dietary amino acids. That's in my case, the focus of my lab, dietary protein restriction, right? And they all have beneficial effects on improving metabolic health. And so research has an easy way to test the effects of diet, right? We get a mouse, we set them in a cage, we give them a certain diet, and we can measure certain endpoints, whether it's on cardiovascular disease, diabetes, neurodegenerative diseases, or what may have you. But the reality is we don't live like that as humans, right? We have buffets of food where we can just pick from whatever we want. Our buffets in Louisiana always look like crawfish and potatoes and corn. But the question here to focus is, are we really eating for more than just calories when we're at these buffets, right? And so animals, they have to find a way to actually sense their nutritional needs. So what makes them do that? How do they sense the need to take in carbohydrates or to take in protein or fats? And we can look at that. We can look at that from the perspective as the hummingbird here, or bears, right? You mostly see bears foraging on berries or honey, but they also eat protein. So how does this nutritional sensing in nature actually work? And what are the mechanisms that could be driving this need for protein? And so before I jump into that, other labs have actually tested where they have isolated macronutrients and compared that to energy intake. So let's just go through this figure here. So if you look at the amount of protein here at the bottom of the x-axis, and you see 5, 10, and 20. So the lower the protein, the lower amount of protein in the diet, the higher the amount of FGF21, and it's stable. And even there's been reports that carbohydrate intake actually influences FGF21, increases in FGF21 circulation, but it is not as closely tight packed or stable as it is with protein restriction, as well as in fat. And energy intake really doesn't have an effect on FGF21 circulation. And so our data from our lab strongly suggests that FGF21 is a signal that is essential for these coordinated responses to dietary protein restriction. I always like to pop in this figure here because it gives us some background to the different types of FGF. So they're endocrine, paracrine, and endocrine. And our main focus is 21. Reason being, once mostly released from the liver, it has beneficial metabolic effects on most tissues, right? So within fat, we can see a reduction in body weight. increases in energy expenditure. It has some effects on food intake within the brain, and that somehow increases energy expenditure. And it also is beneficial for lowering blood glucose levels, as well as insulin and triglycerides. So yes, we know FGF21 is beneficial on metabolic health. And so the Morrison lab actually tested this question back in 2014 and 15 by taking a diet that was low in protein and low in protein of 4% and compared it to a normal protein diet about 18%. And we, where fat is constant and the carbohydrates are a little bit more increased, but not that much to affect the levels of induction of FGF21. So here you can see, at early as day 4, you see an increase in FGF21 in the low protein fed animals. And this exists out to day 14. And these are in wild type mice. You also see a reduction in body weight, an increase in food intake, and an increase in energy expenditure. But when you give the same low protein diet to FGF21 knockouts, you see that this response to dietary protein restriction is blunted, and it's actually no different. It's no different compared to the FGF21s on the control diet. With dietary protein restriction, you also see an increase in thermogenic regulation in white adipose tissue, increased UCP1, as well as CEDIA, and you also see the induction of multilocular cells in white adipose tissue. So we have some evidence that it's actually, this white adipose tissue is starting to brown, but you do not see those effects in FGF21 knockouts when they're fed a low protein diet. And so that goes back to the question, where is FGF21 actually acting to induce these beneficial metabolic outcomes? And so it was actually shown in the Mangelsdorf lab that if you delete the beta-carotein receptor out of the brain using the CAMK-CRE model, that you do not see a reduction in body weight change, but you actually see these animals that have intact FGF21 signaling in the brain. When they're giving FGF21 back to the brain by ICV, you see this reduction in body weight change. And they also showed that this actions on the body weight change is an effect of increased BAT SNA activity after the ICV injection of FGF21. But there are other reports that show that FGF21 acts in the fat through dependent and independent mechanisms. So these are adiponectin KLB knockout animals that are deficient for FGF21 signaling in the adipose tissue. And as you see here, if you give them an injection of FGF21, they respond similarly to a wild type animal that received the FGF21 injection. However, their whole body glucose intake is only improved when FGF21 is functional in brown adipose tissue. And so the question that I was faced with when I joined the Morrison lab was, where is FGF21 acting in response to protein restriction? We know protein restriction has all of these beneficial effects on metabolic health, but where is it actually acting? So our model was to test dietary protein restriction. We know it increases FGF21 from the liver within the circulation. And does it act in the fat or does it act in the brain? One way to do this, we deleted the beta-carotein receptor, which is the co-receptor for the FGF21 pathway to induce this intracellular response. And so what we used was just a CRE model, and we were able to knock out KLB in fat as well as in the brain, the hypothalamus, so it's knocked out everywhere. So this is just a confirmation that we were able to delete KLB within the white adipose tissue and the BAT, as well as in the hypothalamus. And so first here I show you the outcome of deleting FGF21 in the fat. Animals that received a low protein diet, they respond similarly, the KOs respond similar to the wild types fed a low protein diet, as well as a reduction in body weight gain. They also increase in food intake, and they also increase in UCP1 within their white adipose tissue. But we also, as I said, we knocked out beta-carotein in the brain, and these animals respond to the induction of FGF21 by a low protein diet. Does it act on the liver to induce? However, they do not. The only animals that showed a reduction in body weight gain over time were the control animals fed a low protein diet. Thus, the brain KOs don't sense, or they did not respond to a low protein diet to reduce their body weight. And it's just the body weight points over time, and this is the change or the gain, which you see this reduction in the body weight gain. We also saw the increase in food intake only in the controls fed a low protein diet. And we saw an increase in energy intake only in the control animals fed a low protein diet and not in the KOs fed a low protein diet. It's correlated with the increase in UCP1 in the white adipose tissue. We also took our same low protein diet and tested whether low protein could be beneficial in the context of obesity. So we used a high fat control diet with normal protein and then a high fat diet with low protein. And as you can see, the control animals and the KOs both respond to high fat, low protein diets on the induction of FGF21. But only the controls for the high fat, low protein have improved glucose tolerance, as well as they have a reduction in their insulin levels, in their blood insulin levels. And so FGF21 signaling in the brain, we've shown that it's required for the metabolic adaptations to protein restriction, but not in the fat. And so FGF21 signaling in the brain induces this physiological, favorable, but adaptive shift in energy expenditure and glucose metabolism, which is beneficial for health. But it still doesn't answer the question, what does this mechanism regulate preference in nature? So animals, they have to sense their need for protein. And we were able to kind of mimic this using mice within the lab. So we test the question, can FGF21 drive the changes in food choice, meaning the protein appetite? So Jamie McClutchan in 2018 actually published a paper showing that if you restrict rats, these are rats, if you restrict rats and you give them a choice to choose either casein or maltodextrin, that they actually increase their preference or their choice for casein. So we decided to test this same question, yet in the context of FGF21. So here's our design. So I'm going to kind of go through this kind of slowly here. So these are wild type animals, and they have a single choice of either control diet. They have a choice of control diet and low protein diet. They're given control diet or low protein diet, and they're given either casein or maltodextrin. So as you can see on day one, the animals that were protein restricted, they increased their preference for casein, but there was no change in malto intake. So we repeated this with the two choice as in the McClutchan paper. And these animals are wild type animals on either control or low protein diet. And they're given the choice of either casein or malto. And you see there's a reduction in the malto for the low protein fed animals, but there's an increase for casein. And so now I'm going to show you the data with testing this FGF21 involved. So we repeat the two choice design with wild types and KOs. And you can see here, as you can see here, that the wild types that were given the two choice that were protein restricted, they decreased their preference or their intake in maltose, and they increased their intake in casein. And so we also retested this in the brain knockouts or the CAMK knockouts. We see a reduction for maltose only in the wild types or the control animals that were protein restricted, and an increase in the casein with no changes in the brain knockout animals. And so we also wanted to test the same question without using the diet to induce this FGF21 signaling. So these are wild type animals fed chow, and we give them the choice to select either casein or maltose. For 2 days, they get a single injection of FGF21. And as you can see here, these are the wild type, the control animals fed a low protein diet, injected with FGF21 actually increased their casein intake on day 1 and day 2 with a reduction in their maltose intake, but there's no changes in their total intake of casein or maltose, but the preference for casein is increased. And so FGF21 signaling in the brain is required for behavior adaptations to protein restriction, and that the signaling actually induces a physiological relevant adaptive shift in macronutrient preference. And so overall, these data represent that FGF21 from the liver to the brain coordinates these adaptive responses to dietary protein restriction. And to this date, no other hormone has been identified to serve in this physiological role. How am I on time? Good on time?
We have a couple of minutes till 9:35.
Okay. Well, I got one pop in after Anna's talk. I was able to pull up some data. I didn't think I was gonna have time to pop it in, but after Anna's discussion on sarcopenia and Matt's question about protein restriction having an effect on sarcopenia, or if it's being identified as a negative therapy or to induce sarcopenia in aging or older individuals. So of course, these are mice, but I recently completed a longevity study. And so I just popped some data in here about lean mass and preservation of lean mass as these animals are aging and being protein restricted at the same time. So of course, this was not unusual for us to see this reduction in body weight gain over, of course, these are 22 months. So they've been on diet since they were 3 months of age. And we actually, the endpoint was at 22 months of age for this study. And this is the end points for body fat. And of course, there's a reduction in the body fat percentage, but there's no real difference in the lean mass percentage within these animals fed either a low protein diet or a controlled diet. We also see that there's a slight increase in grip strength, and this is normalized using the lean mass. So maybe Anna, we could talk. I do have some muscle tissue, and I could share it with you if you're interested in at least the wild types. So please let me know. And with that, I would give my acknowledgements to my advisor, Chris Morrison, and everybody in our lab. Some names here, Heike Munzenberg, she's an advisor on my F32, as well as Jacqueline Stevens and Michael Schwartz, that's there with you guys. Some of our collaborators are Steve Simpson and Sam Salon Bayet in University of Sydney, and some other professors at Pennington that are always willing to give advice and mentor, Hans Rudy and Tom Geddes. Steve Cleaver was a big help with providing us the CAMK knockout mice for us to breed. Dr. Ingram and Andre Barkey, I will forever be in debt to Dr. Barkey. He's my graduate advisor. And I would not have gotten to the aging field or geroscience field without him. And so with that, I would like to take your questions or comments, or you can leave them in the chat box. And thanks again for having us.
All right, thank you so much, Cristal. It was fascinating. And it's now 9:36, so I would say that we are officially on our 35-minute coffee break until 10:10 when we're going to start the next session. There is one question for you in the Q&A tab, Cristal. If